Hello and welcome everyone to today's seminar about From Target to Market, an overview of the industrial process of drug development and possible job profiles. Our speaker today is Uwe Lohmeier from the Gläsernes Akademie in Berlin-Buch. I'm really happy to welcome you to VBio's first soft skills seminar in English. I'm Simon Häusler. I'm from VBio, the Munich office, and I would like to introduce VBio to you in a very brief few slides. VBio, that's Verband Biologie, Biowissenschaften und Biomedizin in Deutschland. And like almost all things in German, that's also pretty long. So the English translation is the German Life Science Association. We are a non-profit umbrella organization for all scientists in the life sciences in Germany. VBio ensures quality standards in studies and careers. We improve research conditions for scientists and therefore advise politicians in many things. We also organize teacher trainings and free online courses like this one. VBio is an independent advisor for decision makers on all issues of biological relevance. To be a member in VBio is only 35 euros for students and PhD students. So have a look at vbio.de, what we do, and we're happy to see you register. Just very briefly about upcoming seminars. So we have one more next week. It's in German again. It's Persönlichkeitsprofile und mögliche Karrierewege. And then we have a summer break until the end of September. And we again start with a seminar in English, boosting your motivation, how to tackle hard times during your PhD. And this we do in cooperation with Franka Schröder. She's also from Berlin, and she's a systemic coach, and she might have some nice tips for you. And then we have 4 other seminars until Christmas. So have a look at master-bio.de for a lot of more information about these seminars. Very quickly, this is a book that we just brought out a new edition of, and it's now available. It's in German. I don't know how good your German is already, but here we have about 90 CVs of scientists, of all of them biologists, and they basically cover all the different fields of careers possible for biologists nowadays. So if you're orienting yourself about your future career, have a look. You can place an informal order at info@vbio.de. And this is also a free welcome gift for new members. And with this, I'm already through. Become part of VBio. We represent your interests in Germany, and you might like to support us through your membership. You can also scan the QR code here, and it will bring you to vbio.de/Beitritt. And before handing over to Uwe, I would like to ask who's in the audience today, and I will just quickly start this over here to ask your professional occupation. Where in your career are you currently? Please just click the boxes, and you can maybe continue clicking those. And I will show the results in a moment. Almost 80 people replied, and most of them, 44% are master students, 27 are PhD students, and 17 are bachelor students. There's also one researcher from Akademie. And there's also 6 postdocs and 1 science professional and 2 clicked others. All right. So you might see the results now. And with this, I hand over to you, Uwe, and I'm looking forward to a very interesting talk. Your audio is still...
Can you hear me?
Here we go. Yeah.
All right. Thank you very much. Okay, so, Simon, thank you very much for that nice introduction and for welcome the audience and myself. I try to open my presentation.
Yeah.
Please let me know whether it works.
Yeah, and one more thing, I forgot about the technicalities.
Yeah.
So I wanted to say this in the beginning, right? So we are using the Q&A function of Zoom to collect all the questions, but Uwe will have a presentation, and it might happen that a question that comes up in your mind in the very beginning might be answered in the very end of the talk. So we're kindly asking you today to maybe have a little text file on the side and make notes what you want to ask. And in case your question is answered during Uwe's talk, you obviously don't need to type it into the Q&A. Otherwise, I would like you to use the Q&A. And if you find questions that are very interesting for you as well, you can also hit the like button and they will go up and we will prioritize them in the end when we have the discussion. All right, your presentation is visible, and now you, Uwe.
Yeah, good. Okay, I really appreciate that Simon takes over the control and all the management, because normally I do that job during the GLA courses and workshops, and I really like to be in the speaker position tonight. So yeah, and therefore I think it really, it should really fun to go through the topics I would like to provide to you. Yeah, and I'm really happy also to answer the question. As Simon said, it is a very, very broad area in which I would like to introduce you. And I found it really hard to bring a clear structure in it, and probably to pick everybody up from your position. I saw that most of you are students in the bachelor position or in the PhD position, and so probably it's a good idea to wait until I am finished with that, and to start then with the question session. Probably during my speech, it may happen that a number of questions will already be answered. So the agenda for my speech, the first introduction, where I come from and where I'm working in. Most of the parts for this speech on industrial process of drug development and possible job files will be, because that is the most information to share, is the overview of the process of drug development. It is, as I said, it's a very complex topic. And so I will raise in the beginning some basic questions. We are in a highly regulated environment here. So to introduce some guidelines regarding the regulatory framework, what is about research and development? We will go very briefly into the preclinical and clinical phases, that you have at least a very thorough overview on which we are talking about, and that you probably have an idea how many-sided the different teams are, and coming with that also, what are the job opportunities in this area. Then we go a little bit deeper into the regulatory framework. We have also the topic, which is not really my cup of tea, regarding the intellectual property, the licenses and the business development, the overarching guys doing or holding the complete team and processes together are working in the project management. Also here, some words about them. The drug safety has to do with pharmacovigilance. This is also a broad area in the clinic, in the pharmaceutical industry, not only in the development part, but also here probably it could be good to have here also some words about that. Good manufacturing practice means the quality control or the quality management system in manufacturing drugs. Yeah, and at the end, from my perspective, in being for many years in the big pharma, some words regarding the soft skills, some pros and cons, and probably to be decided by you if you probably plan or considering to go or to make your way probably into the industry or to pharma. And yeah, and if you want to go deeper into the processes, I will also introduce our summer schools. Really in these topics there which we will have this year later. So my goals, as I said, we are talking here really of a very, very huge topic: clinical development, drug development, and corresponding job profiles. So I must do at first an excuse here. I can provide really just a very rough orientation in a very complex network of processes. If you are not already familiar, and this can sometimes really be confusing, it has a number of variations and a number of correlated functions and relationships here. So I hope it will help a little bit to clarify your questions, but not to to enlarge the confusion of this topic. That would be my aim of this talk. So please understand that with my given examples here, that I describe only the standard scenarios, and keep in mind that always exceptions are possible in almost all corners and functions and steps. My idea is in that 1 hour speech at least to create at least a feeling for the huge complexity that anyway bears a great potential of individual development, that you get a little bit of thought of that. And I would appreciate if this speech would motivate you to dive a little bit deeper into the broad, really broad field of drug development. And maybe you will derive some fun to join comprehensive following workshops. There are a lot of them, and also lectures, webinars, congresses. So if you dig and if you go into the internet, you will see a number of different courses and and options on that. Many thanks before I start. I must make to my trainer team, which is active in our summer schools from the GLA, which are Wolf-Stefan Richter, Michael Hildebrand, Michael Viergens, Matthias Schröter, Oliver Ladendorf, and Michael Buchholz. You will see in the later slides in the footer quite often from whom these parts are from. And I derived a number of slides from the slide desks from our summer schools. And I'm really thankful that I have this huge reservoir to compile this overview here. my trainers or the trainers of the GLA summer school, they are actually the real experts and practitioners in their specific areas. And so please understand that I probably in the question session afterwards, I would definitely not be able to ask or to answer all of your questions, but they are able to provide the complete picture and advice, as I said, probably also here in our summer schools. Yeah, about myself. I just came across that I got my diploma in biology last year, exactly 30 years ago. I studied biology at the Freie Universität in Berlin, and I worked here on the chemistry and lichens. So I'm a botanist, actually. And I did my PhD in plant pathology afterwards at the Technical University in Munich. And then I started already when I completed my PhD thesis already in 2001 to 2015 in Big Pharma. And I started here at the quality unit in the clinical development at that time at Schering. And 2006, there was the merger and Schering Pharma became Bayer. Exactly from 2001 to 2007, I worked as a clinical auditor. So that means I worked in clinical assurance in the clinical development area. From 2007 until 2012, I with own project responsibility, I worked, so to speak, as a senior global clinical auditor at the global study audit management function within quality at Bayer. That is a job I explain a little bit more later on, which has to do with a number of cross-functional work, with a number of travel work, with a number of cooperation with several functions. So after being almost 12 years in that area as being at quality assurance, I changed within the quality function to the global process management. That is more an in-house job where I dealt with the procedures, with the processes. Probably you might have heard about the SOPs or the WPs, the standard operation procedures or the working procedures. Yeah, and since 2016, I changed completely my employer, namely to the Gläsernes labor at Buch. And since that time, I worked at the Gläsernes labor Akademie, the GLA management, and sometimes also as a lecturer for the school classes. And since 2017, I am actually the head of the Akademie of the Gläsernes labor. Yeah, so in pharma, in the drug development, actually, I worked for almost 14 years. It is already a while ago, but I hope that is still enough to describe the complete process and the complete mechanisms. Some words regarding the Gläsernes labor Akademie. That is the unit on the campus, on the Gläsernes labor, offering business orientation and practical training in life sciences. We are part of the Campus Berlin-Buch GmbH. That is an economically independent non-profit company located in the utmost northeast of Berlin. And this GmbH is managing the research campus as a whole. And to the GmbH belongs also the Gläsernes Labor and therefore also the Akademie of the Gläsernes Labor. The GLA, as I said, is offering career orientation programs for doctoral and postdoctoral researchers in the life science area. That is the one branch. The other part is because we are working together with a number of really good scientists here on the campus and also from externally to provide an advanced training program also for the technical assistants. Since February 2024, we started also the talks in the Cube. In our new Berlin-Buch, which is an incubator, the brand new building on our campus. And with these talks in the Cube, we want to bring together the business and the science and to connect founders of startups or life science companies together with the scientists. And that worked quite well since 2024. We are actually in the summer break and will continue in September with the next topic. We have overall experience in the life science education and training programs for almost 25 years. And we have many-sided collaborations, first with our institutes on the campus, which is the Max Delbrück Center for Molecular Medicine, the MDC, which is the Institute for Molecular Pharmacology. This is the FMP. And we have also cooperations with BPI, the Bundesverband der pharmazeutischen Industrie, with the TÜV, with other parts from the industry. And this brings me here also to this format here together with the BPI. And we cooperate since many years very successfully together with the BPI, and we set up also a course, a summer school, many years ago with the BPI. I really appreciate that we have highly professional trainers and lecturers who are really open to share their expertise and practical knowledge in research, development, and business. And our idea is really that we bring together those experts with our trainees in a relaxed but high quality courses, because in this combination we learned that the training effect is very high. And and therefore I think it's, and I appreciate that I can really considerable influence this program. And whenever we have new scientists on board who are willing and who bring new ideas on board, I'm always free and I'm always able probably to add their topic, to add them into the program. And that is really fun here. So in consequence, we are able in our program to offer one-day workshops, the already mentioned summer schools, or intensive specialist courses. Yeah, this is a little bit regarding where I'm come from and what I'm doing here. So, and now we can really go into the topics for today. So if you are interested in drug development, you have to raise a number of questions and you have to be aware about a number of questions. The first very important one is, what do you need to develop a drug? Of course, you need, or you have probably as an excellent scientist an idea. When you are in a company or when you want to build up or start a company, you must take care that your intellectual property is safe and is secured. drug development is a very expensive area, so you need a lot of money, and it is questionable whether you as a company, probably as a startup company or as a private person, has enough money. So you need an investor was rich enough and interested enough to spend money for that. And of course, you want to develop and you want to exist further. Probably you have the idea also to become rich. And therefore, it makes sense to get also the money back, at least sometimes in the future. So it would be a good idea if your substance is successful. And being successful means to get a marketing authorization. And and to get the marketing authorization, there must be an authority who says, yes, I'm convinced your data are fine. All the rules were adhered to, and I'm convinced that this is something which has a benefit for our country, for our people, and therefore I give the marketing authorization, and it may be described, it may be sold, and then you get probably the marketing authorization back. So this is a very basic but very important question you have to raise. To go a little bit more into detail on these questions, to differentiate between company, investor, and regulator, so the different positions are, as a company, you must be clear on that. How would you continue the development? You must know what is important and which steps do you want to go, or do you want to build up? You must also know that the investor wants to know what would you want to go before you invest in a company to give money, and what would you expect from the company. And the regulator, for them, it's interested to know what you want to, or to know what you need to know before you give the marketing authorization, and under which circumstance you would probably refuse to give a marketing authorization. If you condense this complete environment, you come to a number of really important questions. So that is, the first one is, what do you want to improve and how? In other words, how do you want to earn money with your idea? And this is triggered by the question, or by the reason: is there a medical need to develop your drug or to go further with your idea? The next one is, you must be able to handle risks in your developments. So that means you should be aware, is the science solid enough that you can base your idea, your development on it? You must be aware of the regulatory requirements, a big area here, because you change from the basic academic science into a high regulated environment here. So there are a number of strict requirements regarding the non-clinical world, the clinical world. We will, during my next slides, we will always see the CMC term, which means the regulated area affecting the chemistry, the manufacturing, and the control. So keep that already in mind, CMC, clinical and non-clinical. To be successful, it might be a good idea to be well aware whether your idea can be early proven for efficacy, or probably also on the other hand, regarding futility. And as a founder, or as an overall to say, as a person who has responsibility, you must also be aware that you need the right people, and that you can also provide your staff also really a perspective, being from the work and also for their life management, because people might have the idea or the requirement not to be with you for a year, but probably also for half a life or even longer. So you have also here a high risk to consider whether your staff is really solid, and whether your staff can also have a perspective in your company. And as I said, the money plays always a big role here. So that means a commercial viability must be given. So that means you should be aware how much money does the development cost, and you will see it will cost really, really a lot, and how much money can be earned. And it must be at least so much as the costs are. And the success and the money to be earned with your idea can depend on the target group. That has also, again, something to do with the medical need. Probably if you have a widespread disease for which you can use your drug, are there already competitors on board? Probably at the time of marketing authorization, probably that can minimize the success. And always during the drug development phase, and keep that please in mind, regulators and the complete process always ask, does it work? Is it safe? So that must always be kept in mind. Is your drug safe? Does it work on the indication you have in mind? And as I said, cannot be underestimated. It is not always on the screen of a scientist or as an original scientist. Also the protection, intellectual property, licenses, all that stuff cannot be underestimated and must be considered. As I said, clinical drug development overall is a very important, is a very expansive topic. Excuse me. From Michael Hildebrand from the Biotech Pharma Summer School, he has compiled for us a very impressive overview here. You see the increase of comparison of full costs per entity from the 17th, from the 1917s until 2013. In the 17th, the cost of bringing a new chemical or biological entity to market was about 180 million. And in 2013, we are at $2,560 million. And the majority of the money goes really into the clinical trial phases here. So half of the money costs really the development part, where you use or where you consider or perform the clinical part of your development. So that means if you go, when you go into with your drug into humans, yeah, that costs the most. That is the highest amount of money. Yeah. That is a very confusing, but also a very comprehensive overview of the complete drug development process here. And this is something I usually like to show that also during the summer schools here, because it makes very clear that a number of that a number of functions are involved here, starting with research discovery over non-clinical development. Then comes the expansive part, said already, the clinical development, and afterwards the post-approval life cycles. And we have here, with these symbols here, a number of decision points, or in the project management, you call it milestones here. And you see that you go into the run with 8,000 candidates of leads, of potential candidates for being developed further and finally to become an approved medicine. That goes down, and that is selected, goes down to 250 candidates within the non-clinical development, and at least when it goes into the clinical phase, you are at least at 5 medicines, and finally, after the proof of concept of the proof of concept stadium, will come later on this. You are here with 1 medicine, and all of the money must be earned back, must be brought back by this medicine. So that is how the complete process is actually working. Yeah. In this overview, you see how many functions are actually involved here. We have here we have here again, with these blue arrows, we have here again the complete process steps here, but little bit clearer depicted here under these blue arrows. You have here the different function here. You see that the research functions are at the beginning here and just only punctually involved and later on. You have here chemistry, manufacturing, and control development that runs overall over the complete process here, the production. You have also over the complete time of non-clinical and clinical development, the questions of toxicology and pharmacokinetics. That means how poisonous is, or is there a poison potential of a development candidate, and the pharmacokinetics clarifies, to overall speaking, the general distribution of pharmaceutical active ingredients into the bodies. All clinical development and operation functions are involved, at least per information, also already from the early phases, but actively involved. That is clear here from the clinical development phases. Reg affairs, we come later on to this, is involved over the complete processes here. Yeah, very important to know, and that gives you the overall picture regarding the involved function here. Most of you, I guess, do work in the academia part, the same as I did before I started at Big Pharma. And you see here in the value chain overview in the pharmaceutical market that the academia is involved mostly, as I said, always as a standard. We have always here also exception cases, but most of the parts in academia working with discovery and and with target identification, target validation, and drug targeting overall, to speak, that is the same as you will sometimes read about drug delivery, means the ability to direct a drug to a desired site of action. And the target identification means a target protein implicated in a disease. And the lead discovery means to find lead compounds for activating or inhibiting targets. Yeah, that leads here to that ADMET stadium here, and that means all the processes dealing with absorption, distribution, metabolism, excretion, and toxicity. Yeah, you see that the big pharma or biotech may also involve or may also have already the discovery parts. So probably if you think about Pfizer or Bayer or whatever, also they have sometimes an own research unit where the people do more or less the same job as the researchers in the drug development discovery part in academia. And biotechs do concentrate usually here also in the discovery part, but sometimes have also some early parts of the drug development here. Yeah. Then it goes further in the parts where the biotech companies, or then afterwards the big pharma companies, dealing with the clinical parts of the drug development. So that means here it comes here to the submission or to the clinical trial application. So that means you start with the first studies in man. When you are lucky, then you probably can submit all your data for approval. That is the new drug application, and the outcome is hopefully then the market authorization. Yeah. Most of the money will be, and that is not, probably most of you know that already, that is not really new, the proprietary marketing. That is where you have really the patent, where you also are patent secured. If you are off protected regarding the patent, then you go into the generic marketing, and in that time here, really the money must be earned. Afterwards, there is normally, with a successful drug, there is no further option really to earn considerable money afterwards. As I said, the complete development process underlies really a high regulated area, and I must spend some words regarding the underlying international process. The history is that of ICH, which means the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human. The background is that regulatory authorities and pharmaceutical industry founded this council here in April 1990 to discuss scientific and technical aspects of pharmaceutical product development and registration, to come to an international harmonization in terms of development. And they formulate and they agreed on technical guidelines and requirements. That is very important, and that is really a success. And I'm always I think it's really strange that it came relatively late. I mean, in 1990, after a number of catastrophes in clinical trial, in doing clinical trials, or think about think about German before 45 and all that stuff. And also to harmonize the approach of doing research in drug development came relatively and surprisingly relatively late. And yeah, as I said, it was originally founded at that time by the member states of the EU, by the United States and Japan. And meanwhile, a number of associated members are there, and they agreed on a number of guidelines. And from these guidelines, they have to be translated, they have to be transferred into national laws and requirements. That is, here you have here the national guidelines. One very important of these ICH guidelines is the GCPE6, talking about good clinical practices. That is the quality and ethical standards in clinical trials, based also on the Declaration of Helsinki. So meanwhile, they have developed a number of guidelines in different areas, and we will see from these areas here, from these different areas, a number of examples later on. However, this is the guideline framework for all states who wants to do clinical trials, which data have to be or are considered to afterwards to be agreed on a mutual basis. Yeah, very, very important. If we go a little bit deeper into these ICH topics, you have seen it here. You see it here above. You have the quality topics. There is stipulated chemical and pharmaceutical quality, talks about stability, impurity, good manufacturing practice. That means how to make drugs. Risk assessment is here. You have here the preclinical stipulations, talks about, for instance, toxicity. You have the pharmacology. Here are covered all the preclinical studies in animals. This is the area about the clinical trials. So that means trials with humans. Yeah, that is because in human trials, it should be proven whether the drug does work. Yeah, is it safe? Does it work? So you need here also to define drug safety, which is pharmacovigilance. As I said, the good clinical practice, that is the quality standard and the ethical standard, and a number of topics more. And Under M, you find here a number of topics which are overarching all on all of the other parts here. Okay, let's go a little bit into the landscape process overview. So raw material should become a drug. So what do you have to do? Which step have to do, and which material do you have to provide? CMC, you remember, that is chemistry, manufacturing, and control. And you remember from the first flow diagram, coming from research, academia, it is also regulated, but it is not the ICH area. Brings in the target and active substance discovery. It has, within this CMC area, a process has to be set up and developed, what to do with the raw material under controlled condition in order to have the material for the preclinical study. You need also a process, a regulated process for stability studies. You need also a process, a validated process for the GMP, that means the good manufacturing practice. And the API means here the active pharmaceutical ingredients production. And this process is needed to have here the formulation, to have here the production, the production process under control. That means if you have a tablet, if you have a syringe, if you have an ampule, all that stuff must be under regulated processes, and all that stuff talks about GMP here. And also here for to safeguard and also to ensure that the GMP process is working, you need also here stability studies where the material comes from here, as well as from the fill and finish process to see whether this is working. And finally, from this formulated fill and finish process comes finally the material for the clinical studies. So here, the production process, you have that is the validated process to be sure that you have safe and verifiable processes and substances to go at least, or to go finally into the clinical studies with humans here. And if these clinical studies are successful, you come finally to a registration, and registration means that you get the marketing approval there. Again, target identification, target validation, target optimization is part of the research early development. Then you go into the CMC area, chemistry, manufacturing, and controls. And you see here the different paragraphs within the ICH regulatory environment. This is one of the Q family here. And another one is here, the manufacturing. That is one of the, that is the good laboratory practice here. Yeah, and you have also to do controls of analytics, the duration. That is something which didn't fit into the diagram on my slide before. The duration here lasts about 2 years as an average. The preclinical phase then contains the process development, always the stipulated pharmacology and pharmacodynamic studies, the safety and toxicology. And here we are, as I said, in the safety area of ICH. And the components you need for this development step is a number of labs which have to be validated, which have to be qualified. That may be the cell line development, the cultivation, the molecular biology. You need in the in vitro parts, you need cell bank storage, and perhaps in most of these steps in that early part, in the preclinical part, you need also labs for specialized analytics. That may be to evaluate mycoplasma, virus analytics, and so on, and preclinical testing labs. And of course, it is easy to understand that a number of other facilities of contract partners are involved. You need also a process of controlled shipment here. This part here is also listed here. The costs are relatively low in comparison. You have that in mind, the numbers in mind for the clinical part, but the time you need additionally here is relatively long, namely about 1 to 2 years. As I said, always keep in mind, this is a standard I'm describing here. You have always exceptions where also these parts are much, much, much longer here. So if you have all these stuff in the preclinical phase under control and that is working and you are fine with that, you have all, you have the authorities made happy, you have your investors made happy, then you can directly start into the clinical phase. Can you? No, you cannot. It is still a very, very complicated picture. And Michael Viergens, our clinical expert from the summer school, I had him recently in an expert panel in the talk in the Cube in the last months. He gave us a very nice but really confusing overview of the complete picture here to confuse you a little bit more here at that part. And the headline is here: the path from preclinical development to the start of the first clinical study can be challenging, especially for highly innovative technology and specialized development products. That is really true, and Michael has a huge experience of these stuff. Here you have already some topics we have already discussed on. You have the idea, you have the funding, you have here the research data. You come in the lead selection. You have here probably you are happy to select a lead candidate. You make some proof of concept studies in vitro and in vivo. You have to find also, if you are not equipped sufficiently here, and this is sometimes also not at big pharma the case, you have also to find some contract partners here. The CDMOs means contract development and manufacturing organization. You have further funding steps here. You have to develop your product formulation. You have to provide the GLP tox studies, and finally you have to come to a clinical study design, and you have also to find a contract research organization, probably, which does for you all the study management. We come to it, we come to this topic a little bit later, and you have to manufacture your investigational medicinal product. This is the abbreviation for IMP. Yeah, and when this is going smoothly, you can formulate and then you can submit a clinical trial application to the authorities and start the first study in human. FIH is first in human. But, and now the but is coming, you may face a number of loops here. Probably your lead candidate flops in a first attempt. Here you have probably to adjust something. Your contract partner are not adequate or do not what you want. That can also be the case with your manufacturing practice. Also here you have to adjust something, and the clinical study design, that is really an own picture, an own procedure for itself. That may also be very tricky and must also be agreed with the external functions, be it the authorities or be it the ethics committees. And this takes also a while until you have everything on start. And yeah, so this is also here a very, very complicated, sometimes also for the staff, a very risky step to come from the preclinical part into the clinical. So that means you change your subjects, so to speak, from animals to humans. You start a trial with humans, and it is absolutely understandable that here you need really the highest regulatory insurance and safety also, yeah. And therefore, and that makes it very complicated, must be done with a number of experience. And as I said, especially for highly innovative technology that may cost a number of money and time, which probably was not foreseen, even if you start up and if you do not have the expertise here. So is there hope? Where can you start? And hope is again in the ICH guidelines. And if you open here the E8, which talks about the general consideration for clinical studies, you find in section 4 the drug development planning. You find here the subsections regarding quality, non-clinical studies, clinical studies, and all the planning aspects regarding these topics. And ICH E8 has also an annex where we just have a brief glance on it, where the types of studies are characterized and categorized, which are, you have the pharmacology studies here, and there the tolerance and safety are evaluated and proven the very first time. You have the pharmacodynamics, the pharmacokinetics. How is the drug metabolized here? It asks regarding cardiac toxicity or other organ tolerances here. You have exploratory studies, so where you describe whether you want to find out whether your drug is doing well for the intended use, for the intended indication. You will have an idea regarding the dosing or the dosing regimen, and probably also a dose-response relationship can be expected here. accordingly, you have here the dose finding studies. You can also explore in this phase also biomarkers in that. And if your drug is surviving these exploratory phase, you come into the confirmatory phases where you hope to demonstrate the efficacy and to establish the safety. Still having in mind, we are searching for efficacy and safety. Does our drug work? Is our drug safe, at least as the other comparable drugs on market? And how to do that? That is a randomized controlled clinical trials. We come to that later on. And if you have that on market, if you have your market approval, you have afterwards another phase where you look into the post-approval phase to refine perhaps dosing recommendation, to also disclose hidden or very seldom adverse reactions and so on. As I said, as an example here, regulatory guidelines or guidance. We have here in the EMA, the European Medicines Agency, formulated also based always on the ICH standards scientific guidelines, as well as the Food and Drug Administration of the US. So that is the corresponding or the responsible authority in the United States, clinical trials guidance documents. So from all the ICH connected countries, you find such of these guidelines or laws which must be known and which must be harmonized with each other when you are doing a multi-center, multinational study to make the complete regulatory landscape a little bit more complicated as it is already. Let's have a look into the drug development program for the clinical phases. We have seen that in the overall picture, it may last in an average between 10 and 15 years. And to define it a little bit clearer, also in the wording of the authorities and within the companies, you have here also some divisions regarding the phases. And I would introduce you a little bit into the phases of the clinical development in clinical studies. First, very interesting or good or to keep in mind is the term IMP. I explained or I mentioned that already. The IMP means any medicinal product which is being tested or used as a reference, including as a placebo in a clinical trial. And the clinical development overall covers the first use of an IMP in humans, often but not always healthy volunteers. You have the first use of IMP in patients. You are covering the evaluation of safety and tolerability. And in the proof of concept, you evaluated the first time the efficacy and you confirm the therapeutic benefit and you provide data for at least the marketing authorization. So the regulator looks into the data, and in the case that the regulatory authority finds the data convincing, you will get the marketing authorization here. In the phase 1, as I said, safety, tolerability, PK will be tested here. You have the pharmacodynamics in the phase 2 studies here. Phase 3 talks about or handles the drug, the dose finding, and the efficacy and safety are in the big phase 3 studies here. To get an overview or to get a thought in which sizes we are talking here, the phase 1 studies, so that means the first time when humans take a research drug, are 6 to 20 as an average. The length per study is quite short. We are talking about weeks and months, mostly with healthy volunteers, because you want to see the distribution in a normal, so to speak, human environment, but also sometimes depends on the indication, may also be possible for patients. Bigger are the phase 2 studies with around 100, 200 people with in a length about a couple of months. There you go already in symptomatic patients to see whether your concept is working and to find the dose. The huge phase 3 studies may include more, a number of thousand people, may last, depends on the indication and the application for years. And there, hopefully, you can confirm your proof of concept here. And as I said, afterwards, the post-approval has a broad range here, also to find seldom adverse events, to have post-marketing commitments with health authorities, and to have, again, here, the seldom adverse events or long-time observations also for the drug safety. The proof of mechanism here refers to the early clinical drug development in phase 1, usually performed in healthy volunteers. Little bit to give you a little bit more information regarding the early phases here, showing that the drug exposure at the target site of action, showing that the drug interacts with the intended molecular receptor or enzyme that has really something to do with target identification, with drug targeting from the very beginning. You have seen that in the research part. You want to have here evidence in humans and showing that the drug affects cell biology in the desired manner and direction. So here to collect as much as scientific information you may or you hope to find, and you hope to be confirmed from your early research phases. The proof of concepts is a small group of patients with disease of interest, phase 2, and showing a useful amount of desired clinical activity. And the proof of principle, this is done here as a phase 3 phase used to confirm what you have found out in your early phases here, and that you also you hope that you also show here statistically significant evidence of clinical efficacy and safety here. And these data from the phase 3 study are usually those which are used also, or which are needed to get the marketing approvals here. First in human study designs. I think it's worthwhile to have here also a deeper glance into the study population. So the pros and cons between patients and healthy volunteer. Keep always in mind we are talking here about the standards. Exceptions are always there. It depends on the indication, depends on the drug. If you have a patient in the study population, patient may benefit on a multiple dose, but keep in mind that the discontinuation of investigational treatment is very likely in those early phases, even in case when the patient does benefit in an early stage of drug development. So maybe the ethical committees does not accept such a clinical protocol with patients in a phase 1 study. The cons is here, as I said, it goes what I just said, a single dose as a rule has no benefit to patients. For patients to be used here, that means it requires a frequently washout of the preceding medication, which is sometimes an unacceptable burden for ill patients. The recruitment rate may be low, so that means that this phase 1 may be long, and time is money also here. Sometimes it means a multi-center approach. So it means you should have a number of hospitals with patients participating in your phase 1 study, and patients with underlying diseases may probably affect your biological parameter profile you want to find out and you want to research on. In the opposite to that, the pros of healthy volunteer is you have a high scientific data quality, and you see a safety profile, and you have reliable PK data in a very short time. The cons, which need to be weighted against the pros, is healthy volunteers take a drug but do not have a benefit regarding their health. They have only risk, and therefore it can be assessed as being unethical or not feasible for certain therapies. Think about oncological patients. The study conditions are not the same like in the real world, because you finally want to give the drug to patients. The cohort size, I said that already, is quite low, differs in an average between 6 and 20 patients. It is driven by the statistical power, you know, probably also from earlier or comparable studies, or even from the animal part. In phase 1, the statistical power is often not possible, and therefore you are limited to a descriptive statistic only. That means mean, standard deviations, and so on. And it must be balanced between the minimum number of subjects on which it is reasonable to decide on dose escalation and the number of subjects exposed to an IMP. Placebo is mostly given. So that means the first in human studies typically placebo controlled. That helps to interpret significance symptoms. So that means flus with caffeine withdrawal, in-house diet, lack of exercise, and so forth. So also to have an arm without the IMP with a lead, that means a placebo. To give a placebo makes sense. That you can distinguish between drug-induced effects from others. Blinding is always a big topic here. because blinding means that the subject does not know whether they got a placebo or a verum, as well as the investigator does not know that as well. So the medical doctor being responsible on site for the study, as well as the subject does not know whether they get a placebo or a verum. It has also a little bit in this part a disadvantage, because pharmacological effects can lead to an unattended, unblinded of the study. You must define additional blinding rules. For instance, to take care that only special stuff like the pharmacist or the PK analyst may need to know the treat allocation here. So it is possible that from your study team or that from your sponsor team, only a part is actually unblinded. A dose selection means that the dose range and plasma concentration that exhibited PD effects in animals, nature of these effects, slope of PK-PD curve, you derive your dose selection from toxicity seen in animals, and you have perhaps sometimes the possibility to have comparative disposition of different animal species. And you have the presence of active metabolites with long half-life, and you want to approach here the starting doses here. A washout period must be considered. So that means you need really people without any treatment, substance in their bodies. Yeah, so you need also to do the right decisions and to do the right things to consider the washouts, and you have to define also stopping criteria in the study protocol. The study protocol is really the defined and approved description how to perform clinical studies from the very first study on until the phase 4. All studies in any steps need a well-defined study protocol with an authority approval and with an ethics approval. Very, very important to know and to keep that in mind. As a conclusion, for the first in human studies, study objective, product type, and ethical consideration must be done. Examples are here biosimilars in oncology and adaptive study designs may consider other study-specific aspects here. And if we go further and if we were successful in our phase 1 studies, we can go into the next part, namely the phase 2 studies, which covers first the proof of concept and the dose finding. So we differentiate between a phase 1 and a phase 2A and a phase 2B here. The exception can sometimes be in oncological indication that a phase 1 can also be used as a trial to be submitted for approval, which is a pivotal study. That is sometimes possible, but this depends also on the indication here. Again, here we are still here in the exploratory phase, in the descriptive phase here, and that proof of concept. And in this proof of concept, you plan that they intended to give a clear indication that a drug is a serious candidate for further investments in development. This is also for business reasons a very important step, because here it is sometimes the reason to come to a go or to a no-go decision, and a go decision means into the phase 3 study, into the phase 3 studies, also to come into the really expansive part of the clinical development. These studies help to optimize the study designs for subsequent pivotal phase 3 studies. So that means also this is the preparation phases or preparation studies, so to speak, for the phase 3. And the conclusions have an impact on the IMP itself and to the entire program for which similar compounds may around. So typical findings here to terminate the clinical development here coming from the proof of concept study, maybe it becomes evident because you are dealing here with patients, really with real patients, that the drug is tolerated only poorly at an effective concentration, or has an unsatisfactory pharmacokinetic or metabolism effects, or has a low potency or even an absent efficacy here. The proof of concept here is you need to be randomized study trials. You need control that is in different variations possible. You need endpoints, and you need single endpoints. That means probably for oncology study, time to progression. You can do here also the biomarker studies for endpoints. As I said, patient populations are quite a few. You need washouts here. And you need also a rigorous safety monitoring. And you also need to here also very clearly defined stopping rules here. Yeah, that maybe efficacy is the outcome of interest where safety is embedded, or you need also, or perhaps also lab values from the kidney or from the heart or whatever. That means the proof of concept phase 2A is key deliverable of exploratory drug development. And that is the early point in the drug development at which the weight of evidence suggests that it is reasonably likely that the key attribute for success are present and that the key causes of failure are absent. Yeah. Some people say this is the step to change from research into clinical development, but this is a matter of definition here. Then we come to the next phase, which deals with dose finding. In those 2B studies needs to be learned the dose-response relationship. What is the effect size the drug can achieve? To identify the increasing part of dose response, because you want to really go with the optimal doses into the next study phase relevant for submission, and which doses leads to an unacceptable efficacy tolerability, and ultimately to choose doses for the phase 3 trial. That is what I just said here. That is also a prepare study to be successful in your phase 3 studies. Again, this depicts a little bit if you are selecting too high doses here, you will probably see an unacceptable toxicity, and you cannot continue with that in further trials. And if you choose a low dose, that will probably show no effect at all in the confirmatory phase. So here can also be the risk that overall the study drug is working, but not in that dose. So here the dose finding study, very important to go with the optimal dosage and formulation into the later studies here. Yeah, and here, as I said, now we are in the last phases here, in the last phases before registration. And you see here in that late study to prove the efficacy of the therapy and to confirm the safety of the drug and to establish really the proof of principle. That means that you have also a statistic relevance, that you have a significant evidence, and that you have a clear clinical efficacy and safety. This diagram belongs here beneath the phase 3 part here, and you see here in this you have a considerable increase of information regarding frequent safety information, regarding efficacy here, that is the red curve here. And you see here already, because you're dealing with a relatively high number of patients, also the occurrence probably also of rare safety information. Yes. Nevertheless, you are here confirmatory, and you hope that your very expensive phase 3 study go together with a huge amount of organization, manpower, capacity, comes really to an successful to successful results. Again, here hundreds and even thousands of patients of many months or even years. You want to see in your primary objective an improvement of disease in your target population, and you need endpoints which are easy to measure and which deliver stable values over time and are reproducible and sensitive also to change. Also, the number of patients involved can be great, because since you have already a number of safety information, the patient risk is relatively high at that phase. And you have also here the large scope of these trials give researchers the opportunity to prove efficacy and overall efficacy as well, also to identify relatively rare side effects of the treatment, if any. Yeah, and also these phases are running mostly in a blinded fashion, also with placebos. The study teams are blinded until the study is over and until the study and until the results can be opened and the results can be unblinded. Or I think we have seen that already, confirm the therapeutic benefit. That is what I said already. And yeah, and these studies, the team hopes really that they provide the adequate basis to get the market approval. That means the study may also further explore dose-response relationships. Also in wider populations, you have also here the dose-response relationship in different stages or diseases, or also in combination with other drugs. Yeah. Nevertheless, if you thought you have done everything right and correctly, and if you deblind sometimes the surprise is very bad, and sometimes the study teams, the stakeholders, the money givers, and the researchers see nevertheless that a big portion of such drugs under development flop and fail. So you see the courses of fail due to efficacy overall from those which are failing are 56% due to efficacy and 28% due to safety. Yeah, that is considerable. And the attrition rates are still high. And the high attrition due to a failure of therapeutic benefit are more than 50%, and safety issues almost 30%. The therapeutic areas with poor overall understanding fail more often. So that means here, because we have a huge research requirement still in the understanding of oncology methods and oncology mechanisms, and correspondingly, the failure of the therapeutic area is mostly here in the oncology part here. Yeah. If you were lucky and you have submitted your studies and your documents with your data and with all your studies and with all your study outcome, and you have the approval and it's on the market, you have still to do the the post-approval studies. That means in the phase 4, to understand the product in the real world situations, to obtain evidence for higher reimbursement or submission for expanded labeling. That is also possible. And probably also the regulatory authority require also a further observational phase. And the monitoring safety of a drug in a larger non-clinical setting, to have really here the real world conditions outside a regulated and world of a clinical study here. So therefore, the phase 4 begins after drug approval. The therapeutic use studies go beyond the prior demonstration of drug safety efficacy, as I said. The studies in phase 4 are studies performed after drug approval and related to the approved indication. And there are studies that were not considered necessary for approval, but are often important for optimizing the drug's use. So that is also in the benefit of the company to get more data to optimize probably the drug here. Involve safety surveillance. As I said, sometimes the authorities, the FDA perhaps, or the EMA, stipulated the phase 4 studies. And we have seen that also sometimes that adverse effects, side effects, only detected in phase 4 studies may result in a withdrawal or restriction of the drug. That has happened. The evaluation is also of efficacy is also here to observe or to find rare or long-term adverse effects, and pharmacogenetics. As I said, healthcare costs, that is also, I mentioned that already in the beginning. It may be important for decision-making, whether a benefit of a new drug is worth the money, and other important factors determining the benefit besides the drug effect is hospitalization, fatalities, overall patient satisfaction. That is, this is tools, these are tools or criteria to find out other important factors. Yeah, to summarize a little bit the clinical phases here again, the phase 1 means the clinical first steps in clinical development with to assess the safety, tolerability, pharmacokinetic, pharmacodynamic. Some phase studies are performed in parallel to phase 2 and 3 to support drug development. We have seen that in a very optimized way when we have come to a very early approval of the corona drugs. Phase 2 to 4 is a transition from exploratory to confirmatory clinical development with the aim to assess efficacy, safety in a broad population, and phase 4 to get also some post-marketing information. The study objective, product type, and ethical consideration determine the study design and other study-specific aspects. Key success factors here is that you are able in your clinical phase to identify the optimal dose here, to have also risk mitigation strategies for first in human studies, also to consider the right indication and sequence, to consider also biomarker, bioanalytical strategy to get the most data and information out of your study as possible. Yeah, and I said, very, very important always to keep in mind the compliance with all regulatory stipulations, first of all with GCP and all the other regulation. Yeah, and also to ensure the comparability after manufacturing changes. That is also possibility. That is also very important. Also to guarantee the continuation of the quality of your study drug here. That was a long part regarding the clinical phases, the preclinical phases, the complete process of drug development. Now we come probably to a topic which wakes you up again a little bit until this part here, the study team setup. That means the job opportunities. So if you are working in a clinical or in a pharmaceutical company, you see you have here a variety of different job positions here. In the center, there is a clinical project manager here, and he or she works together with a number of different other functions, be it the quality assurance, the auditor. This is where I worked for a long time. The medical writer here, writing together all the data here, working together with the data manager. The statistician belongs to the study team here. You need an analytical unit here. You need the study management team here. Very important is the regulatory manager and the clinical research associate or the monitor. That is the hot wire or the interface to the clinical study side. The clinical study side is when we are dealing with patients, with humans. Only medical doctors, physicians can perform or can do clinical studies on behalf of the sponsor of the biotech company. So you need an investigator and the team of the investigator, and the interface, the communication between the clinical study team and the sponsor is the clinical research associate. Very, very important here. Yeah. We are already quite far in the time. I got a little bit the clock out of my views. Therefore, I will short up a little bit this scientific advice from the EMA, FDA, from the national authorities are always on board. So you have also to cooperate very close with the authorities from the very beginning on. So that means here also the regulatory affairs, the authorities in permanent discussion and in permanent exchange of the data, of the results. You as a sponsor, you as a pharmaceutical company, probably as a biotech company, has to prepare and have to be in permanent communication and in data exchange with the regulatory affairs. So the corresponding documents you have to submit permanently are the investigational medicinal product dossier, for the investigational new drug. This is an IND. You have also the investigational, you have that also for the clinical phases here. And finally, you compile together the dossier for the marketing authorization in close cooperation with the authorities. Very, very important. It is easy to understand that the dossier containing as a big part of the CTD of the common technical documents contain a huge amount of information which has to be compiled, which has to be checked for inner consistency, for verification, for validation. So you need also here in the reg affairs team a huge staff which has the oversight, which does the work here. Just to give you a thought about which documents we are talking here about, and you see you have all the preclinical parts here, you have the clinical parts, all that stuff. And you see here, as I said, the different countrywide and national organizations, authorities. In Germany, we have the BfArM here, or the Paul-Ehrlich-Institute when we are talking about cell therapeuticals, or when we talk about vaccines. For most of the other things, the BfArM is responsible. In the United States, as I said, that is the FDA. Under the umbrella for the European medicines agencies, the authorities in Europe work together, and many other drug authorities in the different countries have to be considered, have to be involved, have to be convinced, have to be in the loop. Yeah, that is a very huge topic. Therefore, it is not, or we cannot already stop here. Just a word regarding the intellectual property in biotech and pharma. As I said, that is not really my cup of tea here. You see also the patents, also the intellectual property has to be considered, have to be on the screen here. Here you have a general overview. When you are dealing in your biotech company with one of these substances here, you must also have an eye on the rights of intellectual property and patents. That means also a number of questions besides all the safety efficacy discussions within the clinical development have to be considered in the business development. Some general questions also here. An economical assessment has to be considered. Yeah, and you need to know what a patent exactly means, what a license exactly means. You have to be aware that a number of license types have to be considered, as well as sub-licensing that is also possible and to be considered during setting up your business plan, and also during also during the business plan examination. For all that stuff which has the oversight here, that is the project manager. And the project manager, that is our project manager, Matthias Schröter from the summer school, makes a joke sometimes, is deemed as a person who thinks 9 women can deliver a baby in 1 month. Actually, the project manager has to remain the full oversight here, has to deal with time, costs, content, and scope to make the stakeholders happy. He leads the project. He or she leads the project, is responsible for almost everything in your preclinical project. Wherever you divide your complete picture and landscape, the requirements is good knowledge of the project management, the general management, the product specific knowledge. He or she must resilient and endurant and has a holistic and sustainable way of thinking. Yeah, that is a little bit where the project manager has to be and to whom the project management has to report. And last, let me just highlight through 3 aspects here from our ICH topic: the good manufacturing practice, the pharmacovigilance, and the good clinical practice here. The pharmacovigilance is, as I said, the drug safety. The definition is here: drug safety is a science and activities relating to the detection, assessment, understanding, and prevention of adverse events or any other medicine-related problem. The EMA coordinates the European Union pharmacovigilance system and operates services and processes to support pharmacovigilance in the EU, as all the other pharmacovigilance units in other countries do the same. Also here, you need a pharmacovigilance unit in every company which is dealing with development, with drug development, and therefore continuous surveillance of safety information is needed. Another process which is joining the complete process from the very beginning of drug development until marketing is the good manufacturing practice here. Also here, you have a number of job descriptions here. You have a number of possible structures in the good manufacturing practice here. And to come to a conclusion here, the soft skills in drug development, pros or cons with question marks here. For me, as I can say from my long years working in quality assurance and got to know the clinical functions and divisions and different companies here, your thinking must be quality driven, very important, because I said it a couple of times, you work in a very high regulated environment. It comes clear when I described a little bit the project management. Wherever you are working in a company, you are a team player here. Sometimes from the upper management, projects are stopped, although they are from the clinical data and from the development successful. Probably management decisions come to the decision that the project is stopped. This is one example, as well as many others. So you must be able to tolerate frustration. Be open, very, very important, always in all directions, because you have always in your companies and even in the especially in the very huge companies, the option to form or to influence your career way in different and in many ways. So also be flexible, also be ready to be flexible, also to change your mindset. Many functions need the ability or the readiness to travel. When you are in the hot phases for compare or to prepare your data to perhaps to be submitted, it can sometimes be very crazy and very and very turbulent. So be resilient also when you came across problems and shortcomings, and always remain curious. That is something you need as a soft skills in addition to be a good scientist or a good wherever you come from. That is I would say these are basic soft skills, and ask yourself, is that a pro or is it a con? Therefore, the question marks here. Yeah, if you want to go deeper in these procedures, in these topics, as I said, our GLA is doing a GMP Biotech Summer School in October, where we dig together with Michael Buchholz, who is an excellent expert in manufacturing, especially in those of advanced therapy medicinal products, with the participants over 8 days. We do that in cooperation with the BPI. We plan and I think we will also offer the basic training to come familiar with the good clinical practice, the ICHE6, you remember, with Doris Greiling and Christoph Ortland. That is a webinar offered by the GLA. As I said, I hope we can offer this, or we can bring this on our program in December. Yeah, and this is the target from target to market. This is our GLA Biotech Pharma Summer School, the next time in September, where you will see the slides, what I have shown sometimes, or some extracts during this talk here from our colleagues. They will dig into the different parts and phases and aspects of clinical development, from the target to get the market approval. As again, this is a course in presence in Berlin from the 10th to the 13th of December at the GLA. Yeah, and that's it so far from my side. I think it costs me, more time than expected, but I hope you have heard some important and interesting information in that big topic. Thank you very much.
Thank you, Uwe, for these valuable insights into the drug development processes. We do have a couple of questions in the F&A. I would say some of them are the very classics that we hear in those webinars more often, because a lot of the people are concerned about their own qualifications and how they can end up in certain areas of this pharmaceutical industry, and they're always interested if you actually need a PhD or not is one of the biggest issues, I think, in biology or in natural sciences, I would say, in general. And what would you say, how mandatory is a PhD to work in the biotech industry? Or do you have certain jobs in your list here that you could say, like, okay, there you could have a master's degree, and here you do need a PhD?
I think the times have changed. I think when I started in big pharma in 2001, I think it was definitely from my position as being in quality assurance in the GCP area, my boss told me I expect to have a PhD there. That was mandatory for him. But I must say at that time, I was really lucky because I had only a very, very rare experience in clinical development. I did a job for a while as a data manager. So I had actually a thought, a clue what a clinical study is. But the big plus was really my PhD at that time. But I would say times have changed, and the possibility to apply also at big pharma, coming direct from the university and to get a good job are much higher than at the time when I applied at big pharma. It is, I would say, it is more complicated, and the probability is lower if you applying directly at big pharma as if you would apply at a small biotech company. Mm-hmm. But do that always when you have finished your studies, also without a PhD. It is always worthwhile to do that and to try. An excellent possibility to get a huge insight and experience, and where you have a good chance to start at the pharmaceutical industry in clinical development is to answer or to respond when you see an advertisement regarding clinical research associate. Definitely, I would say the times are so that definitely a PhD for this is not mandatory. However, if you work a while as a clinical research associate, you have really a wonderful first foot in your company to go further, and probably also to go into the study project manager, into the study management, and in a number of other into other jobs. The jobs wherever you need medical expertise, just medical advisor, or for the pharmacovigilance. Therefore, a medicinal study is mandatory, and also here I would say by most of them also a PhD.
Okay, to the clinical research associate, but a master's degree is the minimum, or can you also go with a bachelor degree with maybe additional qualifications, or without?
I would try, and nevertheless you have also at the big CROs, and sometimes the big CROs are even bigger than the pharmaceutical the pharmaceutical companies themselves. They have internal academies and schools where you can go with your master or with your bachelor degree and apply to be developed and to be schooled and to make your exam, your internal exam as a clinical research associate. That could be a way, also being a bachelor.
Okay.
Yeah.
Okay. And then one more about the qualifications. So how is it after someone spent about 4 to 5 years in academia as a postdoc and doesn't have any publications? Is this a problem, or do you need publications to go into any of the study team?
No, definitely. That I would say definitely no. I think publication cannot be bad, but normally they do not support. What actually really support, and at my time when we had the applicants, they have sometimes really wonderful long publication list, and my head said, okay, it's nice, but what can you tell me what helps and support being a clinical auditor here? And we were always very happy, and I think that's still the same. If you have done additional add-on training, be it having joined the biotech pharma summer school or the GMP, the GMP summer school or any other additional workshop training on clinical development. That is really a plus if you add this certificate to your application documents. That is, I think, that weights more as a long, wonderful publication list in the pharmaceutical industry. Very, very important and very good question. Yeah.
No. Okay, thanks. And then for the project manager especially, what is the straight way to reach such a position? Which concrete steps can you take when just finishing your master's degree?
As becoming a project manager, I would yeah. Yeah, good question. Also here, I think you can ask yourself whether you have already some project manager experience. It can really be in a completely different area, probably also in whatever concert organization or whatever, or in the cultural area, or when you or restaurant management or whatever. If you have some experience in doing project management and really, as I said here, the general management knowledge, the endurance and resilience to be very communicative. And so if you have some evidence in your CV that underlines that you have here some specific experiences in being a project manager, do that in your CV for your application. Or if you have also a certificate or whatever, or working confirmation or whatever, add this to your application documents. Very important, and that may probably help also with a master or with a bachelor, also to start the development in the company as becoming a project manager. Yeah, I would do that. Yeah.
Okay, thanks. Then there was the question if the recording is going to be posted. And yes, you can find it in the chat. It's going to be on master-bio.de tomorrow, and we'll put it on the YouTube channel of VBio tomorrow. Then one more question. To what extent are preclinical toxicology studies still carried out by the pharmaceutical companies themselves instead of being outsourced to contractors?
Depends. Depends really on the company profile. Depends really on the specific company. Some do, some others do not. It's really regarding their structure, regarding their
size,
organization size. Yeah, definitely. Yeah.
Okay. And which city or state in Germany is currently the leader in the field of pharmaceutical biotech?
Munich, Berlin, I would say. Yeah.
Yeah, Frankfurt maybe.
Yeah, probably. Yeah, number 3. Yeah.
But those 3, I think.
Yeah.
Pretty well. All right, and then the last question. Oh, that's a good one. Which is the best way for applying to an internship as a graduated bachelor student? And just as a background note, maybe not only start applying for those internships when you have the degree already. A lot of the people that we have as guests talking here, they always tell us hands-on experience is one of the best things you can have as an applicant. So starting your internships during your studies is always a good way. First, to see different things, what might be good for you and what not. And then also, some people, they go to a position, I don't know how many years later, and then there's going to be connections you cannot foresee. So always get those internships. But which is the best way for applying?
Go onto the, I would say, go really simply onto the websites of the companies, be it biotech company or be it big pharma, because at the moment, I think the request or the requirement to get good and young and motivated staff is there, and therefore you will see a number of of offers in the internet. So I would definitely look in which company profile, in which size I'm interested in, and go into the internet and to check where are the contacts, where are the corresponding sections and divisions, and just ask them, to call them, to write them, and be proactive, and do your research on the company and ask.
I think that's very good advice.
Yeah.
Would you say that would you say that the chances are bigger in like small startups or in big companies? Because startups might not have the money to pay a lot of people yet, but still need the manpower.
Yeah, definitely, Simon. That is 100% correct. I think to get a good job with much work and less payment, I think the probability is higher at biotech and small biotech companies. And to start with a higher income, with a higher payment, but is bigger at the big pharma, but the probability to get the job there is also lower.
Yeah.
Yeah.
And just another note on this one is that, sometimes there's there's so-called biotech cluster development agencies. Sometimes I can, for the Munich region, I can type this into the Q&A now. It's biom.org. Sometimes, the in the federal states, they have their own resources, and they try to help biotech companies to settle in their states. And this biom, for example, in the Munich metropolitan area, is a really great resource, because you can get lawyers, they help you find lab space and stuff. And they also have a job space usually. And then you can find locally posted job offers that might not be on, the big ones like Job Vector, or even you wouldn't find their website. So this is a way where you can find companies that are still in incubators, startups, something like this. All right.
Yeah, that is also existing for Berlin. That is the Berlin Health Capital website. Also there, I think if you are searching for Berlin Health Capital BHC, you will see also there a number of links, and also a pharmaceutical sector, and probably you will also be successful in finding job opportunity there.
Perfect.
Yeah.
I'll just put it in the chat. Here we go. All right. Yeah, and we do not have any questions open. Thanks again,
Good.
Uwe, for your time and your really interesting insights into the drug development processes and all the job opportunities there are. Thank you for coming to the webinar, staying until the end. And it's almost prime time. I hope you have a nice evening, and see you soon. Bye-bye.
Bye-bye. Thank you very much.